Hello, everyone. Thank you for being here today. I am Pierre Montegruel. I am a radiobiologist in Antwerp, Belgium at the Iridium Network, and I'm very happy to have you here today to talk about flash radiation therapy, this very hot topic now in the field. I would like to thank the ESTRO and also S.I.T. Sardina for organizing this exciting symposium on bridging the gap from preclinical to clinical application in flash radiation therapy. We'll have 3 speakers today who will give 10 to 12-minute speech, all of them, and then we will have questions after their speeches, so please keep your questions for the end. We will have Philippe Portmans, Alicia Gasparini, and Marie Dutrex, who will talk about flash radiation therapy today. We will start with Professor Philippe Portmans. He's a radiation oncologist at Iridium Network and a professor at the University of Antwerp, Belgium. And Philippe today is going to talk to us about flash first clinical applications, a revolutionary approach. Philippe, please.
Thank you so much for the kind introduction. Let's start the presentation. So indeed, I'm Philippe Portmans. I think most of you know me. Okay, this is working. And these are the 2 starting slides, but the title of my presentation is indeed the first clinical applications. And will it be an evolution or a revolution? We will have to see, but there is quite some likelihood that there will be an R before evolution. I have a conflict of interest related to this topic. Let's give a short introduction. Well, we started with a pre-meeting course on Friday. There were 5 pre-meeting courses. We had around 40 people, which was very good also compared to the other courses as a started. This was about intraoperative electron radiation therapy. Just to tell what the flash effect is, the flash effect is that the NTCP, normal tissue complication probability slope, shifts to the right, means higher doses, while the TCP, the tumor control probability, remains identical. This is the flash effect compared to, of course, conventional dose rate. Well, in clinical perspectives, now we started this symposium with first the clinical perspectives, then what we need in physics, and then what we require to be able to start. So it's a little bit strange. It might appear to be the other way around, but I think this makes sense as well. So to translate from preclinical research to clinical research, I dare to say that an adapted machine, an adaptation of a current machine, might be okay for a proof of concept. But the next step needs to be to define all the parameters, the pulse width, the pulse repetition frequency, the pulse height, which dose per pulse, fractionation, all those things that will be very difficult, if not impossible, to manage with an adapted, non-dedicated machine. So with the electron flash machine, on which Alicia will give more information, we have a dedicated preclinical research system that allows full control of all the parameters that we need to evaluate. But the next step is then, what could be the first clinical application? Now, intraoperative electron-based radiation therapy seems to be a very attractive one. Why? Because we could, one by one, do the same thing as we are currently doing, but just changing the time that you give the dose. Now it's typically between half a minute and 2 minutes. If we change from conventional dose rate to flash dose rate, this would be divided by a factor of 100 to 1,000. So it seems to be the most logical next step. This machine, which will be installed next year, second half of next year in Antwerp, and will be available with CE marking early 2013, will be very similar to the current intraoperative radiation therapy machines, but not just changing the dose rate, also taking care of the current shortcomings. It will be much more mobile, it will be much more image-based, there will be a treatment planning system coupled to it, It will therefore also facilitate the introduction of intraoperative radiation therapy in hospitals, clinics who are not doing it currently. Dimensions, weight are fully compatible to put it in a normal room. You don't need reinforced soil, and there is no additional shielding required. So similar to the current machine that we operate in Antwerp, there is just a beam stopper underneath, and depending on supplementary measures, you might put 1 or 2 mobile LED screens to accommodate for the patient-evoked scatter. 8 degrees of freedom, dedicated TPS and automated image-guided docking will be the components that add to the current machine apart from the flash dose rate. If we go to the 6 guidelines from ESTRO ACROP that have been published on intraoperative radiation therapy, and we start with what's done most by those who do intraoperative radiation therapy, it's partial breast irradiation. Current challenges are local tumor control, which is related to coverage of the target volume. The big challenge that has been demonstrated with the Elliott study is not replicated in a lot of others, mono-institutional and other data, is local tumor control because of poor coverage of the target volume. Too small diameters, not image-based, etc. So this is for sure a challenge. Side effects is not the primary challenge. We expect that with flash dose rate, we might safely be enabled to give larger doses and in higher risk patients, larger volumes to be treated. And side effects should with this not increase. Then radiation therapy for the breast as a boost. Local tumor control is not a challenge, but here side effects, local fibrosis, impact on cosmetic outcome is a challenge. So with flash intraoperative radiation therapy, we can maintain the volume dose parameters, but have lowered risk for side effects thanks to the flash effect. For the intraoperative radiation therapy indications in rectal cancer, the challenger are a mixture of locoregional tumor control and side effects. Intraoperative radiation therapy with flash should enable to give the same dose or a higher dose to a larger volume while sparing still normal tissues. I think my first clinical application would be for re-irradiation for patients who have recurrence, and only thereafter in the other indications. If we go to pancreatic cancer, the big challenge is locoregional tumor control, apart from distant metastatic disease, of course, and we are limited by serious side effects of the neighboring organs. So again, flash intraoperative radiation therapy could facilitate volume and dose increase while maintaining, or for lower risk patients, lowering the risk for side effects. So here we could speak of a possible game changer. Sarcoma in extremities, local tumor control is rarely, if done in a dedicated sarcoma center, local tumor control is not a real challenge. Side effects is it more. With flash, we could rather go, because of the thanks to the good tumor control, to maintaining dose of volume to lower the side effects. Retroperitoneal is a mixture of local tumor control and side effects challenges. Also here, volume and dose individualization increase could be feasible with flash effect to maintain or even lower the risk for side effects. And then the new ones, the new possible approaches, high-grade brain tumors, intraoperative. The big challenge is local tumor control, and if we increase dose, we get into trouble with side effects. So again, with flash intraoperative radiation therapy, we might increase the treated volume and the dose while maintaining or even lowering the risk for side effects. And then, not with intraoperative, but with the same tools, skin cancer, non-melanoma. Local tumor control is not the real problem. Side effects to some extent, but especially inconvenience. I find it such a silly image to see a patient with a 5 millimeter skin cancer that is treated with a big external beam accelerator, and the next patient is an S.p.A patient for pancreatic or prostate or whatever. It just doesn't make sense, and it gives a lot of inconvenience. With FLASH, we might maintain dose and volume because local tumor control is not the issue, but lower the side effects and improve inconvenience also because we could increase hypofractionation. Now we give 18 times 3 or 8 times 5. We could go to 5 times 8 and maybe even shorter fractionation schedules. Another one is, of course, the melanoma. First trial will be explained in the next symposium, ESTRO by Jean Bourris. The challenges are a mixture of local tumor control and side effects, dose volume increase, and maintaining the side effects. So if we look at cancer incidence, but more important, mortality, I put a circle around the current indications for intraoperative radiation therapy. The game changer here could be pancreatic cancer, and the bread and butters could be the breast, and then in between the colorectal. If we go to very high energy electrons, the benefits are a finer resolution intensity modulated radiotherapy compared to photons. It's a bit more compact and cheaper compared to protons, and it is very easy to make, it's more logical to make the transition to FLASH, but the challenges to be solved are big biological, technical, and for radio protection. But here, we might be able to have this machine developed. The first plans are being done already. 1.5 year physics research has been done on a very compact machine. Think about something similar. I'm not allowed to say brands, but it's a lookalike from another machine. Very high energy electrons, FLASH and conventional dose rate, 2 models, 50 and 100 MeV. Why 50 and 100? The 50 enables it to do within the current regulations. For 100, it's another difficulty, another administrative layer. Conventional and FLASH dose rate, it will be a pencil beam delivery, of course, in 2D, not in 3D. And it will be compatible to be installed in a standard bunker, not in a dedicated bunker. So coming to conclusion, in 6 years, only 6 years, a lot has been done, but a lot remains to be done. FLASH radiation therapy increases the therapeutic index, which facilitates delivering higher doses for the challenges of tumor control and lowering side effects in cases that the late toxicity is the issue. But we need more studies on tumor control, define the impact of all the parameter settings, and I saw the presentation of Jean Bourris for the next session already. He will give some glimpse on possible evolution there, as will be Marie Dutrex in this symposium. Fundamental biological mechanisms are poorly understood, and we need to share our protocols to work together and to think about the clinical irradiation schemes. And last slide, this is the typical tumor control, normal tissue toxicity. This is the therapeutic window. This is for the mean patient. But there are patients that have a very broad therapeutic window. There are patients that have sensitive tissue and sensitive tumors, and there are patients that have resistant tumors and resistant late effects, normal tissue. So every patient is different. This is a publication about GARD, genomically adjusted radiation dose, where patient per patient you're going to prescribe an individualized dose, which is another thing for the future. And with this, I would like to thank you for being here and for listening. And as I said before, the discussion is at the end.
Thank you, Philippe, for sharing your amazing opinion on clinical application for FLASH radiation therapy. We'll go now for a more physics talk with Professor Alicia Gasparini from the Iridium Network in Antwerp, Belgium. Alicia is a medical physicist. She's also a visiting professor at the University of Antwerp, Belgium, and she will talk to us today about what are the essential characteristics for a FLASH preclinical research platform. Alicia, please.
Thank you, Pierre, for the introduction. I'm glad to be here discussing the essential characteristic for a FLASH preclinical research platform, or the wish list of the physicist. This is my conflict of interest. So potential FLASH radiotherapy currently comes in different modalities. We can see here in this picture UHDR, ultra-high dose rate beams compared with the conventional beam, which having different the radiation type and pulse characteristics. And it is thanks to the combination of all partners involved that we can effectively think of create preclinical FLASH platforms that are used in the preclinical world, at least first. So let's have a look at what, in my opinion, are the essential requirements for these systems. As was already mentioned by Philip, tunable beam parameters. I mentioned this study from Pierre, where they observe preservation of memory above a certain dose rate in mouse irradiated brain, mouse irradiated at the brain, mouse brain irradiation. Yes. Here we can see that above a certain threshold, 100 above 30 gray per second, there is no significant difference in memory preservation or memory loss from the control group. Here instead, we see that from above 30 gray per second, the memory loss is significantly different from the control group. And here's, they nicely reported all the beam parameters. So we can see, and I mentioned the instantaneous dose rate, the 30 gray per second threshold, but here I reported all the different parameters that they used, and they all vary. So it is further investigation is needed to understand what the role of all these parameters is. But this is clear that the beam structure is playing a fundamental role in defining FLASH radiotherapy. So those are the parameters that are selected from this publication from Wilson et al. To be essentially reported together with the preclinical study. So we need to be able, as a physicist, to change these parameters, to systematically change these parameters in a reliable, robust, and reproducible way. So the choice of parameters are those highlighted in yellow. Those are not all independent, of course, and in the following slides I will be talking mostly about dose per pulse and instantaneous dose rate, but the interplay is there. So we want to vary the dose per pulse and the dose rate systematically by changing a number of parameters with our system. We want to do this in a reliable, robust, and reproducible way. And we want to be able to irradiate in both standard and UHDR modalities with the same system. And for the electron flash, which is the system that we are using, we can do this by changing the pulse length, the pulse rate frequencies, the pulse amplitude, but only with the manufacturers. Then we can change also the field size by using applicators from 10 to 120 millimeter in diameter and the distance from the source. And we can do this both in standard and UHDR modalities. Then we would like to have independent and non-destructive beam monitoring. Independence is also a requirement for medical electrons accelerators that we can read here. But it's also important that it does not interfere with beam output, flatness, symmetry, and energy. And here we can see the pictures published of the system that is used at S.I.T. And of course, this consequently will ensure safe radiation delivery because it will be able to stop the beam in case of wrong irradiation. We want to investigate the effect of different radiation types, yes. Not that we want to do this in the same system, but we want to be able to have a system, a dedicated system dedicated systems that are able to be compared with each other. Not sure how yet, but... And this is because it has been shown that different radiation types, here we can see for protons, electrons, photons at different energies, both are showing the flash effect or not. On the same line, we would also like to create a system. We would like the system to support us in creating a common language for reporting, both in biology and in physics. And here is an example of how a misleading definition of a high dose rate, in this case with a dose rate of 10 gray per second, resulted in not observing the flash effect, which was then placed into context by this lettering response to the study. But also for dosimetric study, we need to create guidelines for standardized reporting of dose and other important commission parameters. I will not discuss this too much, but it's important to have to be able to talk about the same dose and the same dose rate when we compare different preclinical studies, and therefore we need guidelines and protocols on this. Radioprotection, we need to comply with the radioprotection requirements, especially in the perspective of using flash, sometimes I go far, flash IRT, but also for EBRT and very high energy electrons. It is important that the system complies with the legislation and the shielding requirements. Here you can see a general study for OR, a placement of an intraoperative radiotherapy system in OR. And this is the part of the study that we made for the radioprotection requirements, the radioprotection of the electron flash from S.I.T. We did it as straight radiation scattered from a phantom, a plastic phantom along the beam. And here are listed the equivalent dose per patient dose is listed in the worst case scenario. And all these numbers are comparable or below the numbers that we could get with a general IRT system within the OR. Those are numbers in the bunker, and those are comparable with the number that we could get for an IRT system in the OR. And then lastly, we would like the system to be flexible to preclinical implementation. This is a larger topic, but I listed a couple. It has to be easy, feasible to be installed in any preclinical facility. It would be nice if we could implement image guidance, and with that we could test and implement also conformal irradiation to see whether it has, it's important, we want to use it or we don't want to use it. Fractionation and study fractionation, different fractionation schemes and volume effects. Also, those calculations could come to a hand. It's handy if the system could be open and flexible for example, multidimensional TPS, where also the time dimension is taken into account. Also, where we could have a plan evaluation by maybe taking into account also the biological effect of different plans, and also to implement real-time online dosimetry. So this is my wish list for essential characteristics for a preclinical a dedicated system. It's open to discussion, of course, and we can discuss it more in depth later. And with this, I thank all the collaborators and you for your attention.
Thank you, Alicia. We'll now go to the biology part with Dr. Marie Dutrex. Marie Dutrex is in charge of the FLASH research. She's a director of research at the Institut Curie, Paris, France, and she will talk to us about new biological data from FLASH research. Marie, please.
Merci. Hello, everybody. So I'm very glad to be here and to more discuss than present data about the biology parameters that could affect the flash effect. And so my conflict of interest. So, well, all of you know that flash was described the first time by Marie-Catherine Vosnin and Vincent Favaudon. And they insist on the fact that if you use high dose rate, you observed reduction of the toxicity of radio-induced toxicity, but you have the same effect on tumor. And reducing toxicity is a challenge for all the oncologists. And the most successful method so far is to fractionate the dose. And it's commonly used. But you all know that when you do so, you also reduce the efficacy on tumors, and you have to increase the dose administered to get the same efficacy than with single treatments. This means that tumor cells and normal cells behave about the same. The new and the revolutionary things with the discovery of Vincent and Marie-Catherine was that maybe now, with a high dose rate, we have a method to discriminate between normal cells and tumor cells. And this is the part that I want to develop with you. So to go back to what we know, there are, since this discovery, a lot of publications have been shown that confirm the flash effect with different devices and different methods. And they were going from, and this is really a brief overview, but they were monitoring very different things with different protocols. The most striking were the study of the toxicity in different organs that has been done in many laboratories. Meanwhile, during all this time, we have publications all the time that describe exceptions. And the big problem we have is it really an exception? So are there really organs that are not sensitive to the flash effect, or specific cells that are not sensitive? Or is it just that the condition where the experiments were done were not the good one to detect the flash effect? And the previous speaker just described the problem of the dose monitoring, of the enough, what you call flash, means what is the dose rate where you see the flash. So all these questions have to be resolved, and we have to not to eliminate the possibility that some of these results are right, but just consolidate this by collaborating together and adding more and more data on it. So, I just wanted to present the instrument we are working in, which is the S.I.T, the first prototype of S.I.T that they designed with interaction with my lab and mainly Vincent Favaudon, where it's very convenient because it has a lot of tunable parameters, so we can really prospect the effect. And, of course, we need the precise dosimetry, and I want to insist that for research you need to monitor, which is something we are not used to. But you need to really to monitor the dose on every sample, every experiment, and not assume that, well, it's about the same, because we are comparing very small differences, and we must be absolutely sure that we are working on the same doses, global doses, and I will suggest to add biological dosimetry in the same time. So what are the biological parameters that could affect the flash effect? And this opens the question, is flash working on any organs, on any tissue? Are there in tissue some specific compartments that are more sensitive or less sensitive? Is it a species difference? I have to remind you that we have very few data about human normal tissue behavior with flash, because it's very difficult to handle it and to do it. So very few experiments were performed on this. So really this is something we have to investigate. In addition, for the biologists we must understand what is why tumors and normal cells don't behave the same. Is it really within the complex of the organs, the tumors, or is it within the cells? That there are some differences that make them behave differently. Is it the state of the cell? They are proliferating of creation, there are stem cells or differentiated cells. Is it their metabolism? Hypoxia is a big issue that a lot of that people are working on. So we need to answer to that. And I just give you an illustration of the kind of study that can be done, and if we accumulate them, we will learn a lot from that. These are 3 completely independent laboratories working on 3 organs with the same question: what is exactly the dose rate effect? So these 3 experiments use varied the dose rate and look at some markers that were specific of the different organs. They didn't use, in brain, it was cognitive markers, so it was a late effect. In lung and intestine, it was more early effect of the damage of the tissue. They used the same dose, total dose. In the first experiments with it's Pierre Montegruel who did it, they varied the pulses. The 2 others, the pulses were fixed data. And they look at what happens when you change the dose rate. The interesting thing is that the profiles are the same. And I think so there are common things that just rise immediately from this study. So we have the threshold where the flash effect, the protection, the difference between the conventional irradiation and the flash appears. Below this dose rate, you see you have no more effect, and after you have a gradient. Higher is the dose rate, better is the difference. So we can wonder, is this threshold the same for any organ? This is, all of them were in the same range, around 100 gray per second. We see we are in flash condition. Below, you may be at the border. And this raises the question of the failure of some people to observe flash when they often use doses that are very near the limit of this threshold. And if the dose, if the calculation or if the dose was not really sufficient, they may be out of. And still we don't know if, because we cannot use the same criteria to measure this toxic effect, we don't know if there is an influence for the definition of a threshold of the test we use and of the organ. Anyway, all the authors observed that the stem cells in the organs were protected in the flash conditions. And this is an important parameter for the biologists because it could explain how you repair after irradiation if you have a good preservation of stem cells. And to further study this hypothesis, we wanted to start some work on human stem cells. So we purified lung human stem cells from patient samples, from normal tissue, from the surgery room. And we compared the effect of flash and conventional on these cells from 3 different donors. And as you can see, we were very happy to see that, in fact, we can detect the flash effect. In 2 of the patients, you see very clearly that at doses that are the dose used in clinic right now, very commonly, we have a protection by flash compared to conventional. However, one of the patients didn't show it. And even more interestingly is the fact that this patient was highly resistant to the irradiation. You can see that with the conventional irradiation, you have much more survival of the stem cell of this patient compared to the two others. So maybe there is a relation between the capacity of the patient to resist to the irradiation and the fact that it is resistant or sensitive to flash. Just one word to say that now we use very commonly one tumor cell line to control the dose. As I told you, this is a biological test. So in the same time you are working with samples, you do tumor cells irradiation, and you expect them to survive exactly the same with your with the two treatments. So we have this cell line and we use it, and this confirmed that the dose were absolutely correct. The next question is, well, if we have a difference between resistant and sensitive to flash, is it the same for the tumors? So we have started to analyze a lot of the tumor sets and to compare their behavior with flash and conventional and their survival. And the top one are the most illustrative. We took long-term tumors. These ones are radio-resistant, these ones are radio-sensitive. And I asked, do we see the same thing than with the normal tissue of a patient? A difference with the sensitivity to flash? And the answer is no. In all the tumors so far we have studied, we have exactly the same survival with flash and conv. So to end up, what we see is that we have individual differences in the ability to answer, to respond to flash, and tumors seem so far to be in the same individual. We have a big difference between the normal tissue and the tumors. Tumors are always not sensitive to the flash effect. This led us to the idea that maybe there are some genetic and epigenetic factors that could be spotted and analyzed, and that could help to understand what is really at work when you irradiate with flash irradiation with high dose rate compared to conventional dose rate. So we are completing this study, and for comparing tumor cells and normal cells, and I just remind you this very nice picture that shows all the difference that transform one cells to a tumoral state in an individual, and why, within this difference, some will be important for flash, and we need to spot them, and we are working on that. And I just wanted to finish also to say that we need in vitro tests, because most of the best results were obtained with animal study. You know that now we have to reduce these animal studies, and in addition, we cannot investigate all the parameters using animals. It's too long and too much animal use. So we have spent a lot of time to try to develop in vitro tests that we hope will be available for the community and will be standard for everybody to test the activities. And this was founded by INSERM and Varian. And these tests will be described on the FRPT meeting, which is a very nice flash meeting dedicated to flash and VHEA that will be in Vienna in December. And I really encourage you to go to this meeting. So this is the end of my talk. Everything was possible because of the commitment of all of the people in my lab. You can see Vincent, who is one of my most active researchers in the lab, maybe the most unpredictable student I have, but he is very inspiring for all of the people. And I thank also all my collaborators, national and international, that are too many to be listed here. And to finish, I will finish maybe like the previous speakers, to say, to quote Euripide, who in 2,500 years before COVID, this is Philip who told me I should date it that way now, said that none of us know what we all know together. So really we have to work together. We are facing big challenge with these new modalities. If we want to go fast, to benefit rapidly to the clinic, we really have to bring all our forces and collaborate. Thank you very much.
Thank you, Marie, for raising these very interesting biological questions. We have 20 minutes for questions and remarks. So for this roundtable, we'll have our 3 speakers plus 2 other participants, Dr. Felipe Calvo from Clínica Universidad de Navarra in Madrid here, and Verdi van Russel from Iridium Network in Antwerp, Belgium. So another physician and a physicist to answer your questions. So please go ahead. Alejandro, please, there's a mic right here.
Thank you very much for the excellent talks. My question maybe is going to be addressed to Marie. It's really evident that to achieve a flash, we need parameters related with it, which is the biological endpoint and the dose rate, any kind of definition of dose rate. My question is related with 2 other parameters, dose and volume. If there is a threshold in the dose, or if there is not a threshold, we should achieve flash in the low bound doses or only around in a layer around the tumor. And the second about volume, if we can irradiate sub-volumes with flash in each one, it should be much more easy. So is there any threshold in dose and in volume?
So in dose, and I hope my colleagues will come because, of course, this is, we have indications that you can detect, well, you saw I was using 2 gray. Say with the stem cells, and we still see the flash effect. So I think one of the problems when people say it doesn't work at low doses is just that you are measuring toxicity. If you are using low dose and you don't have a sensitive test to see the effect, well, you say there is no effect. But in fact, I don't believe that there is I think that flash is working for low doses. High dose, I'm not sure that you can do. Well, no, it's not a miracle. So there are doses where the toxicity will be not overcome by flash. So we have a window where we can work, and past this window, we will go back to a regular toxicity. And this will have to be really defined, and it may depend on the organs you are targeting. As everybody knows, the limit of toxicity varies from one organ to another organ. So don't believe that we can double the dose and have no effect. This is certainly not the case. For the second question was more tricky. Volume in preclinic we cannot really answer because it's very difficult to irradiate specifically small volumes. In addition, we are in animal models. So, we study on the wall. We see flash effect when you do whole body irradiation, when you do whole thorax irradiation, whole brain. But anyway, these are small volumes compared to the human size. But we have no indication that there is a limit in volume. In biology, of course, there are technical limitations of providing a large field irradiation with the instrument, and then you have to move to pencil beam and that kind of things. But for this, of course, has to be demonstrated and checked. But I would say there is a upper limit for the dose, and for the volume so far, we have no indication. Do you agree?
I do agree. I do think that the volume is a big challenge. Maybe, I don't know if Philippe, you want to talk about the volume. IORT will, if you have something to add on that.
Indeed, for both, IORT is typically a single shot. When we switch to the skin with the same machines, we can fractionate. Jean will show the experiment that's ongoing in Lausanne. There it will be single shot and very high doses. But we need to do fractionation experiments as well. For sure, when we make the transition to other approaches like VHE and protons, that will not be logical to then maintain it for larger volumes to single shot treatments. Yeah. Indeed.
I have an online question. Maybe I think you answered to it during your talk, but we can maybe precise. Great presentations. Which clinical applications except skin do you see for electron flash?
So which clinical applications apart from intraoperative and skin? The new intraoperative might be brain tumors. It's, of course, a subgroup of the high-grade brain tumors where open surgery is done as part of the procedure. The intraoperative machine easily is applicable with energies to 12 MeV. I think you can reach maybe 70–80% of those resectable cancer patients. And then the others, then we speak more in the field of the protons and the very high energy electrons. Theoretically, every indication, especially if we prove a flash effect at 2 gray. But let's get practical. If you look into some diseases where currently we have extreme low rates of toxicity, you have to wonder whether that should be your first goal. It could be a goal, but it's not your priority. We have to prioritize. And we have to first look for the challenges, like I made it. Either high complication risks or low locoregional tumor control. And based on that, we can go for many indications and theoretically everything. But that's theory.
We have another question online, which is more for proton flash, but I think we can enlarge it to electrons. If a center can provide a dose rate above 40 gray per second, can they already claim that they could do flash proton therapy or any kind of flash radiation therapy, or do they also need a radiobiological verification of the flash effect? That is a big question.
I'm able to say one word on protons because I just started and I'm learning a lot. We need radiobiology in protons for everything. So we as clinicians, we have very little faith in the radiobiology that is written in the books. We are learning patient by patient, indication by indication, and this is a very tough way to grow. And if you put there flash, well, then we have a big problem, because we have to start from the very beginning. We have to do pilot studies, step by step, very small numbers, very strong observation of the clinical behavior and so on. So flash in protons is terrific, it's technically possible. I understand all the excitement, but we clinicians, we don't have any excitement yet for that. We would like to see in vitro, in vivo, animals, and then moving to flash. And if I can make a comment on Alejandro's and Philippe interventions, we will revisit intraoperative radiotherapy. It's going to be a very nice renewal of all the evidence, strong evidence that we have that is a very efficient technique. In terms of volume, we have evolved from large fields to the tumor bed in sarcomas to do large fields to the tumor bed and a second field to the area of high-risk margin. So it's field within a field. That is already a given in most of the sarcomas, retroperitoneal sarcomas, extremity sarcomas, field within a field, so that you can, and that would be a way to explore subvolumes with flash as well. The second field is always close to the vessels and the nerves, so you will have there an interesting something to look at in terms of normal tissue tolerance. And in terms of those, to tell you the truth, the ones who have been involved in intraoperative radiotherapy for years and years, we will love to move from boosting plus external to full dose intraoperative radiotherapy any site in the anatomy. So many recurrences, many patients that have a margin, a positive margin with a primary tumor. If you don't really need the neoadjuvant chemoradiation, in those patients we will prefer to do full dose and nothing else, not only in breast but also in pancreas, in many other sites. So yes, FLAS is bringing an opportunity to change the volumes, the volume thinking, and to change the dose thinking. Yeah.
Thank you.
I would love to add something, but first, Felipe Calvo, for those who don't know him, and I think that will be few among you, he's one of the icons of intraoperative radiation therapy, co-chaired the pre-meeting course, and he is also director of a proton center here in Madrid. What I would like to say is, indeed, if we just change the dose rate as single factor, you could wonder, does this require redoing everything from bureaucratic and regulatory aspects? I don't know. We will have to see that based on our radiobiology. And then secondly, if we look to parallels, the younger among you will not remember, but in the 90s, we switched from low-dose rate brachytherapy to high-dose rate. And suddenly it was a big panic. It was not, you could not translate it, notwithstanding the experience in some other centers. So then they mimicked the low-dose rate with the pulse dose rate, something in between. So that we have a parallel in the past. And we know that we cannot just give the single shot 50 gray with low dose rate over 5 days to a high dose rate single shot 50 gray. That we know, common sense. So we are fractionating now and adapting the dose. And I think we have to take into account the lessons from the past for the future.
Indeed. Thank you, Philippe. Just a remark from Professor Dirk Verharen, who could not make it today. But to continue on the previous question about the dose rate, as shown by Alicia, it is not sufficient to only state integrated dose rate. There are many more parameters that might be important, and they need to be reported in the publications to help the community to understand the flash effect. We have another question, please.
A couple of years ago, the working mechanism behind FLASH, the indication was that it was bleaching of oxygen, so inducing direct hypoxia in the tumor. And it has a huge impact, of course, if you compare experimental tumors and human tumors. I don't hear it anymore in your presentation. Is that still the working mechanism? Are there other indications of what's the basic indication, according to you, of FLASH?
Well, I think there are, well, the answer is we don't know. There are two models. And we have indication that maybe these two hypotheses apply. One is that with the FLASH, you have a reduction in temporal and spatial reduction of the oxidation you create, oxidative damage you create in the cells, and you may have recombination between some radicals, then delimiting the number of radicals that will attack the other target. The other hypothesis, which is also very well known and has been shown to work in some models, is that you induce a transient depletion in oxygen when you do the flash irradiation. And then you convert your cell in hypoxic condition, which for me, as a biologist, the problem is that none of these hypotheses explain why we discriminate between tumors and normal cells, because the hypoxia status of some tumors is even higher than normal tissue, and they still not respond to flash.
Isn't it that most of the flash demonstrations are experimental tumors, and those experimental tumors, they are characterized by hypoxia?
Yeah, exactly.
If I can add something, Marie, I think now measurements have been done. So I will say I have been responsible for this transient hypoxia hypothesis. It's been proven that we were wrong. I mean, models and measurements in vivo have proven that the transient hypoxia is not strong enough to explain the flash effect. I think it's clear. Models and measurements, at least 2 types of measurements have shown it. So maybe it could explain partly, but the hypoxia induced by flash irradiation is so low that it could not be the only explanations. Doesn't mean that the oxygen and the production of reactive oxygen species is not important, and probably we have to look into that. But the transient hypoxia is not the only thing, and that is for sure. We were wrong.
I would not say you were wrong. It's not the only explanation. And probably it's a combination of several explanations, and it's not the main explanation, the transient. And so Vincent is doing a lot of work to demonstrate that recombination model is, and actually he has very interesting data about that. And, but maybe somebody will come with another hypothesis. We are just a little bit short to really explain how it works. But it's open.
We still have 3 minutes for other burning questions, maybe. Yes, Alejandro? Yeah.
I wanted to have all the rights. This is much more related with clinics. If we have a discrimination between normal cells and tumoral cells, that means that we have a biological boundary. Should it be possible with that to increase the margins?
Yes.
Yes and no. It depends whether we need wider margins. It depends, of course, on the tumor biology, the behavior of the tumor locally, regionally. And I told it yesterday to somebody during a contouring workshop that we now give 50 gray locoregional to the breast, 50 gray equivalent. Sorry, I stopped giving 50 gray in 25 many years ago. But the 50 gray equivalents, why the recurrence risk is not uniformly spread? We will probably, but a bit more knowledge, in 5 to 10 years, start giving 50 gray to the intermediate high-risk zone with a boost to the high-risk zone, and maybe 40 gray equivalent to the intermediate low zone, and maybe 30 gray equivalent to the low-risk zone. And we have already some data, like import low, which was not only comparing whole breast with partial breast, but there was an arm between, where the whole breast received 4 grays only less, and the partial breast was given as a boost significantly less toxicity already, with 4 grays less. So I think that in the future we will more and more give different dose gradients based on recurrence risks.
Thank you, Philippe. We are getting close to 2 p.m. now. If you have other questions, I'm sure the speakers and roundtable participants will be happy to answer to them by email. I would like to thank the speakers for their amazing talks and the participants to the roundtable as well. I would like to thank you for your participation today, and take care. See you soon. Thank you as well for ESTRO and S.I.T. Sardina for organizing the symposium.